Hello everyone. So today I am going to finish the topic ICH. So as per the last video I said I will cover the ICH part 2 in my next video. So this particular video is mainly concentrating on ICH part 2. That is where I'll be mainly concentrating on the stability protocol for the drug substance. And we will see some important definitions which will be useful. So let's begin with stability protocol for drug substance. So if I want to study the stability of a drug substance, then I need to go through some of the protocol which we are calling it as an stability protocol. So before starting, I need to just differentiate between drug substance and drug product. Drug substance is nothing but your API, that is active pharmaceutical ingredient, and drug product, that is what the marketed formulation you are receiving. So let's begin with. So under stability protocol, first is general, that is applicable for everyone. Next, stressed testing, selection of batches, container and closure system, specification, testing frequency, storage condition, stability commitment, evaluation, the last one, statements or labeling. So I'll go in details one by one, starting from the stress testing. So stress testing, it is also known as force degradation study. Stress testing of the drug substance can help to identify the likely degradation product, which can in turn help to establish the degradation pathways and the intrinsic and the natural stability of the molecule. Why we are going for the stress testing? Stress testing is nothing but a degradation study. We want to see that how our API is getting degraded. Mainly we want to discover we want to find out what is the possible pathway of degradation and what are the degraded products and what is natural stability of the molecule. Stress testing is likely to be carried out on a single batch of the drug substance. It should be included that effect of temperature and humidity where appropriate oxidation and photolysis of the drug substance should take place. So mainly stress testing. Stress testing means it is a force degradation study we are calling it as. Means we are trying to give the extreme temperature and humidity and we want to check that how much stable our API is. And along with that we want to develop its route of degradation and what are the possible degradation product. So we are exposing it to higher extreme temperatures and humidity where oxidation and photolysis of the drug substances can take place. Selection of batches. How we are going to select the batches? At least 3 primary batches of the drug substance and should be representative to quality of the material used for the production scale. So at least 3 primary batches we are going to take, and what are the material we are going to use for the production, we need to check for that particular material. Next, container and closure system. The stability studies should be conducted on the drug substance packaged in a container closure system that is the same as the packaging proposed for storage and distribution. Container closure system, that means where we are going to keep that final product, that is drug, not drug substance, we are talking about drug product, where we are going to keep it, which type of container and what is the closure system. We need to check with that particular container and closure system what we are going to use. Next, specification. Specification is depend on what is the specification we have received for that particular product. So specification is a list of tests, reference to the analytical procedure and proposed acceptance criteria is expressed in ICH guideline Q6A and Q6B. So specification, that means what's the specification we got? Suppose it has a limit of microbiological limit, it has a limit of analytical limit. So we need to check it is coming within that limit or not. That is called as specification. Testing frequency. Now, frequency of testing. How we can go for testing the drug substances. What is the frequency? So frequency of testing should be sufficient to establish the stability profile of the drug. It is one of the very much essential criteria to establish the stability profile. First one, for the long-term studies, for the substances with a proposed retest period at least 12 months. For the long-term studies, the frequency of testing, so frequency of testing should be every 3 months over the first year, every 6 months over the second year, and annually thereafter through the proposed retest period. So first, if we are going for a long-term studies, so first 1 year 3 months, that means first 1 year we are going need to go for 4 tests, whereas second year 6 months, that is 2 tests, and after that is from the third year onwards annually once through the proposed retest period. When we are going for the accelerated storage condition, a minimum of 3 time point, there is a 3 time point, that means 3 times, including the initial and the final time. Suppose we are going for the study for a 6-month study. So first we need to start with 0, 3 and 6. That is called as 3 point. Next, for intermediate study, where suppose our study is for the 12 months, there we need to check 4 time point. That means 4 time we need to go for the test. So first is on 0, when we have finished the product, made the product ready, then after 6 months, then after 9 months, and then on the 12 months. So this is called as testing frequency. Now storage condition. What are the storage condition we need to give for our product? What we are going for getting permission from ICH. So we have made it into 3 different tables. First, storage condition in the general case. Next, drug substances intended for storage in the refrigerator, and the drug substances intended for storage in the freezer. First, in the general case, for the long term, temperature can be 25 degree plus minus 2 degree centigrade, and humidity, relative humidity should be 60% plus minus 5% if it is storage for 12 months. And in case of intermediate, 30 degree centigrade plus minus 2 degree centigrade, whereas relative humidity 65% plus minus 5% for the 6 month. Or accelerated, in case of accelerated, temperature is 40 degree plus minus 2 degree centigrade, relative humidity should be 75% plus minus 5% for the 6 month. Whenever we are intending for the storage for the refrigerator for the long term, that is for 12 months, we need to keep it in temperature of 5 degree centigrade plus minus 3 degree, whereas in case of accelerated, we are going to keep it for 25 degree plus minus 2 degree centigrade for the 6 month. Whenever you are going to store the drug in the freezer for the long term, that is 12 months, we need to keep it in minus 20 degree centigrade plus minus 5 degree centigrade. So this is all about the storage condition of your drug substance in general case, refrigerator and the fridge. Next point is stability commitment. That means we need to propose a shelf life for our drug product. In case where data submitted for registration do not cover the proposed shelf life, it is necessary to give commitment to continue the stability studies post approval in order to firmly establish the shelf life. Shelf life is quite important. If during proposal, if you are not giving its shelf life, at that time you need to give a commitment that after once the approval received, you are going for the study for the shelf life. But when submission includes long-term stability data from 3 production batches covering the proposed shelf life, a post-approval commitment is considered unnecessary. So 2 cases, 2 ways it is possible. If you are not giving its shelf life once before while submitting your commitment, submitting your proposal, then what you need to do, you need to give a commitment that after once you get the approval, you are going to study here, you are going to go for the stability studies for the shelf life. But if you have already submitted for the long-term stability data of 3 production batches, then there is no need of such commitment. Now evaluation is one of the important parameter. We need to go for the evaluating our result. The purpose of stability study is to establish a retest period for the drug substance and the shelf life for the drug product. Retest period for the drug substance, that is nothing but our API, and the shelf life for the drug product or the formulation we are going to get. For future based on the evaluation of the result obtained from chemical, physical, biological and microbiological test. So shelf life will be mainly based on the evaluation of the results obtained of different kind of results. That is, it may be chemical results, it may be physical, it may be biological, it may be microbiological test. So generally it should be within the last we have seen the specification within the limit. Based on that we can decide the shelf life of a drug product and the retest period of a drug substance. So for that evaluation we need to adopt a systematic approach in the presentation and evaluation of the stability information, which should include appropriate results from the physical, chemical, biological and microbiological tests, including particular attributes of the dosage form, for example dissolution rate for the solid oral dosage form, along with the which type of dosage form we are designing. Fine. Physical, chemical, biological, microbiological test data should be available, as well as which type of dosage form we are designing. That dosage form attributes we should consider for the evaluation purpose. The next point is statements or labeling. So statements or labeling, as we know that all the drug product will have a label. So what's the label information? How we can give that label information? Where main thing we are considering as we talk about stability, it is in stability guideline. Mainly we are going to tell about storage condition. A storage statement should be established for the labeling in accordance with the relevant national or regional requirements. This statement should be based on the stability evaluation of the drug product. There should be a direct link between the label, storage statement and the demonstrated stability of the drug product. An expiry date or expiration date should be displayed on the container's label. Based on the stability, obviously the expiry date will come. So there should be resemblance between the stability data what we have received and the expiry date what we are labeling in the container, final container. That is called as statement or labeling. So this is all about how we can go for the stability protocol for a drug substance, started from general, stress testing, selection of batches, container enclosure system, specification, testing frequency, storage condition, stability commitment, evaluation, and finally statements or labeling. Now we are going to see some few important terms which can be useful in the coming days. First one, formal stability studies. Formal means you need to include long-term, accelerated and intermediate studies undertaken on primary or commitment batches according to the prescribed stability protocol to establish or confirm the retest period of a drug substance or shelf life of a drug product. Formal stability studies, it can include long-term, accelerated, intermediate studies as per the protocol prescribed, mainly for the retest period for the drug substance and shelf life for the drug product. That is called as formal stability studies. Next, another term, that is impermeable container. Container which will not allow the passage of gases or solvent. Example, sealed aluminum tubes for semi-solids, sealed glass samples for the solution. That means they should be impermeable to the gases or the solvents. Next is called intermediate testing. Intermediate testing, they have some specific temperature and humidity requirement. Studies which can be conducted on at 30 degrees centigrade and 65% relative humidity, designed to moderately increase the rate of degradation or physical change for a drug substance or drug product which is intended to be stored at a long term for 25 degrees centigrade. Intermediate testing is, suppose I want to store a drug product for 25 degrees centigrade. For that, our intermediate testing should be, temperature should be, requirement should be 30 degrees centigrade, whereas relative humidity should be 65%. That means we are going to put that particular drug substance or the drug product in an extreme condition to check its stability, because we are going to store it for the long term. Next, long-term testing. Stability studies under recommended storage condition for the retest period or shelf life proposed or approved for the leveling. Long-term study, that means we need to undergo stability studies as per requirement or as per recommended storage condition for the retest period or the shelf life proposed. Retest period generally for the drug substance and the shelf life for the drug product, which is we are going to propose or it is already approved for leveling. Bracketing, one of the important term. Bracketing means we need to put a bracket from a lower side to the upper side. That is the lowest possible values to the highest possible values. If we come into the in case of suppose a container size, suppose lowest size is 5 gram and highest size is 5 kg. So we need to go for the testing of 5 gram as well as 5 kg, not that 10 kg or 15 kg, 20 kg, not like this. So lower and the higher, that is bracketing between lowest value and highest value. So it is design of a stability schedule such that only samples on the extremes, only samples, that is samples from the lowest one and the higher one, extreme of certain design factor such as strength, container, etc. are tested at all time points as in full design. The design assumes that the stability of any intermediate levels is represented by the stability of the extremes tested. So if suppose I am testing for 5 and 500, so in between whatever the result we don't need, we need to go for the study about 5 and 500. That result what we are going to get, that will represent the intermediate levels, but make sure that we are going for the complete, that is full study design, which I are calling it a full study design. Now come to the next term, that is matrixing. It is also another important. Matrixing means suppose we are going for an 10 batch of production scale. So 10 batch today, tomorrow again 10 batch, day after tomorrow again 10 batch. So what we need to do, 10 batch subset we need to take. Suppose from 10 batch, 1 batch we need to go for the stability study for today, next batch another 1 batch, like this. So if we go and do the stability studies for 1 batch, that is representative of all the other batches. Matrixing is the design of a stability schedule that a selected subset of the total number of possible samples for all factor combination would be tested at a specific time point. At a subsequent time point, another subset of samples for all factor combination would be tested. The design assumes that the stability of each subset of the samples tested represented the stability of all the samples at a given time point. So we cannot no need to go and test in each and every time. 10 batches taken, go for 1. Full study design. A full study design is the one in which samples of every combination of all design factor are tested at all time. Full means everything. Reduced design. Reduced design is the one in which samples for every factor combinations are not at all tested at all time point. That is reduced. We are not going to take all the samples. We will take some few samples. Now next term is new molecular entity. What do you mean by new molecular entity? New molecular entity, if we talk about new molecular, active pharmaceutical substance not previously contained in any drug product registered with the national and the regional authority concerned. It may be a new salt, it may be an ester, it may be a non-covalent bond derivative of an already approved drug substance. That means which is not contained in any drug product. New molecular entity we can consider which is all no one have reported about that. or no, it is not included in any of the drug product. Suppose we can consider as an example of diclofenac sodium. So fine, diclofenac sodium everyone is using. Suppose you are coming with a new salt of diclofenac, that is diclofenac potassium. So we can consider diclofenac potassium as a new molecular entity. As our till date diclofenac sodium was used, we have brought diclofenac potassium. So it can be salt, it can be ester, or any non-covalent bond derivative also. Next important term, pilot scale batch. What do you mean by pilot scale? When we talk about pilot scale in the practical term we call it as it is a connection or interlink between the R&D and the production scale. Once when we talk about R&D, we go in the little bit in the gram scale where we developed something. Pilot scale, it is like a representative of a production scale. Little bit higher than R&D, but lower than the production scale. So here a requirement they have given, generally a minimum 1/10 of that of a full production scale or 1 lakh tablets or capsule, whichever is larger. So this is its limit. So a batch of a drug substance or drug product manufactured by a procedure fully representative of and simulating that to be applied to a full production scale batch. For solid oral forms, a pilot scale is generally minimum of 1/10 that of a full production scale or 1 lakh tablets or capsule, whichever is larger. Pilot scale, as I am saying, that it is an interlink between R&D and the production. Mainly pilot scale, it is like an replica of an production batch. So these are few terms. So this is the end of this particular topic. Hope you will like it. I'll come up soon with a new topic.